The Thermo CentriPAK bioprocessing container system can help make it easy for you to meet the rigorous quality control and assurance standards of your manufacturing applications. From reducing cross-contamination risk during cell culture separation to high-efficiency harvesting and centrifugation, these single-use containers enable high throughput while eliminating the need for post-use cleanup. In this video, we'll take the CentriPAK bioprocessing container, or BPC components, from box to bench. We'll review the unboxing and setup process and share some tips to help you get the most out of your new CentriPAK system. When the CentriPAK BPC arrives at your facility, take a few moments to inspect the packaging. If you notice any damage, please make a note of it on the shipper's packaging list or receipt. Open the box carefully and make sure all the BPC components are accounted for before you discard any packaging materials. Call Thermo Fisher Scientific customer service if any items are damaged or missing. CentriPAK BPC consumables are packed and sealed in two separate outer layers in a Class 10,000 ISO Class 7 facility. You may also choose from various accessories to enhance your CentriPAK system. These include centrifuge bucket adapters to provide a secure fit throughout the cycle, an adapter rack, hanging rack, and more. The CentriPAK BPC is also designed to operate with the Terumo SCD IIB sterile tubing welder. This compact device can rapidly seal and weld tubes that have a 1/8 inch inner diameter. For your CentriPAK system, use any pump with a head that accommodates tubing that measures 1/4 inch inner diameter by 3/8 inch outer diameter. To begin setup, place the CentriPAK BPC adapter rack on a floor, bench, or cart. Set the CentriPAK BPC adapter inside the rack. Put the CentriPAK BPC chamber inside the adapter with the purge container folded in the side basket. Several configuration options are available for your CentriPAK. They include a CentriPAK manifold, which is fully enclosed and sterilized, and a steam-through adapter that connects to your stainless steel bioreactor. There are 3 ways to connect the CentriPAK manifold to the bioreactor. Your first option is to use the aseptic connectors. On the BPC side, attach the tubing to the manifold by inserting the aseptic G 3/8 inch hose barb couplers into the ports. You can also connect the manifold to the bioreactor using MPC quick connectors. On the BPC side, attach the tubing to the manifold by inserting the 3/8 inch coupling bodies that lock into the bioreactor ports. Your third option is to connect using the sterile tubing welder. Once your connections are secure, place the entire system on a zeroed balance while checking to see that all the clamps are completely closed. Check that the BPC clamps are closed first. Now open the purge container clamps and the manifold tubing clamp. Let a small amount of liquid drain to eliminate any air in the system. Next, close the purge container clamp and open the BPC clamps. Fill the bioprocessing containers until the total weight reaches approximately 10.2 kilograms. When you achieve your desired weight, close the manifold tubing clamp to stop the filling process and wait for the pressure to equilibrate. The CentriPAK will automatically balance the weight once the pressure has equalized. To disconnect and seal your bioprocessing containers, use the sterile tubing welder. Leave the manifold connected to the bioreactor and make sure all the connections are sealed and closed. Then, locate the vinyl cap that was included with the components in the box. Fold the tubing close to the seal and place the cap over the fold to secure. Tuck the tubing around the inside of the adapter close to the liquid. Ensure that the tubing with the capped end is inside the adapter and above the liquid to avoid puncture or deformation of the film. If you are filling 8 bioprocessing containers, connect 2 lure locks or quick connectors to the bags before you begin. To flow in samples using quick connectors, first position the CentriPAK BPC under the biosafety cabinet. Insert the tubing cap and plug into the 2 holes near the chamber handle. Pair the BPCs by weight to balance the centrifuge load. The pairs should weigh within 125 grams of each other. Load the filled BPCs into the centrifuge, placing each pair into opposite spaces in the rotor. Adjust the centrifugation parameters according to your protocols. For example, when using 2000 milliliter swinging bucket rotors, we set the speed to 3,900 RPMs or 5,374 times G, the time to 20 minutes, and the temperature to 20 degrees Celsius. After the centrifuge has completed its cycle, untuck the tube first. Then remove the bioprocessing bags gently and place them on the hanging rack. You can connect up to 6 chambers at a time to the Thermo Labtainer manifold. This includes the tubing that connects to the pump. If you use a CentriPAK BPC single with MPC Quick Connect, you can attach only one chamber and its pump tubing to the Labtainer. Now you are ready to aspirate the supernatant. Use the pump to aspirate one BPC at a time. Before you begin, close all the clamps except the one connected to the pump. BPCs can be aspirated while hanging or resting on the adapter. When 75% of the supernatant has been aspirated, stop the pump and use the CentriPAK BPC clip on each bag to separate the pellet from supernatant. After the supernatant has transferred from the CentriPAK BPC chamber to the Labtainer BPC, disconnect the Labtainer and the chamber. To resuspend the cell pellet, connect the Labtainer containing buffer solution to a CentriPAK BPC chamber. Circulate the solution and massage the chamber to resuspend the cells. You can then collect them in the Labtainer. After you have removed the supernatant cell pellets from the BPC, safely dispose of the system components according to your company policy. The Thermo CentriPAK single-use bioprocessing system otherwise requires no post-use cleanup. With its convenience, versatility, and efficient operation, CentriPAK makes it easy to maintain manufacturing compliance while protecting the integrity of your samples.